I am honored to be here. Really, it's always a privilege to talk about topics near and dear to my heart, and I hope near and dear to all of your hearts, since we are all living the mystery of human life. It shouldn't come as a surprise to anyone in this room that recent years have seen renewed assault on human dignity. Human cloning is only banned in about 8 states, and human stem cell research in 5. Assisted suicide is legal in 5 states and in the District of Columbia, where I'm from. People seriously debate in prominent medical journals the notion of after-birth abortion. Or should we be able to decide when babies live simply based on our values? Again, there's a serious and ongoing debate to revise the dead donor rule, defended by people who write articles entitled things like "Death is in the eyes of the beholder," rather than in the eyes of the person dying. And neonatal euthanasia is actually a protocol in Europe that, again, people are trying to adopt here in this country. Assisted dying for healthy older people. One has to seriously ask the question of, is this a step too far? So this is the culture we live in. And it raises 2 challenging questions regarding the nature of human life. So when does the life of an individual human person begin? And this will bring us to the continuum problem. And when does human life end? Which is, I think, the converse of the continuum problem, the persistence problem. So what I'd like to talk to you about today is outlined here. We're going to start with the concept of what is an organism, because I think this is a critical concept for understanding both of those questions. We're going to spend about half the talk discussing when does human life begin, and then the other half talking about when does human life end. But we're going to start, as I said, with this idea of an organism. So the concept of an organism is actually surprisingly not a very simple one. If we consult Merriam-Webster, they give us 2 very straightforward definitions. 1, a complex structure of interdependent and subordinate elements whose relations and properties are largely determined by their function in the whole. Or 2, an individual constituted to carry on the activities of life by means of organs separate in function but mutually dependent, a living being. So if you're like me, I kind of glaze over when people are reading definitions. But the two key concepts that both of these definitions emphasize are that it's the interaction of parts. To be an organism, you have to have parts. They have to have different functions, and those functions all have to coordinate together, work for the good of the whole thing as a whole. Okay, so that's a nice simple definition we can get our arms around, but it's also a pretty simple one. So you can get much more complicated than that. If you really want to go a deep dive into the concept of an organism, I can strongly recommend this paper. It's about 85 pages long, and Bernd Roslingbrock identifies 10 very complicated intersecting features of organisms. So I'm going to try to hit that Aristotelian golden mean here, somewhere between Merriam-Webster and Roslingbrock. And I've divided his 10 characteristics into 4 groups. So organisms show development. They show growth towards a characteristic, mature state that functions throughout that entire process as an integrated whole. They show autonomy, which is self-directed activities and or repair of injuries such that, or in response to perturbations of those activities, such that the unified function of the entity is preserved. They show adaptation. So adaptation and autonomy are related. Autonomy is the ability of an organism to adjust its internal processes to disruptions from the inside. But adaptation is the ability to adjust your internal processes in response to perturbations from the outside. So if you face a changing environment, circumstances that would be incompatible with how you're currently functioning, you adjust your functioning. And integration, which is the very feature that Merriam-Webster focuses on, the coordinated function of parts to support the function of the entity as a whole. Now, I would note that these are discrete, observable biological traits. They're not mystical qualities, and they're absolutely not matters of opinion. They exist or they don't, and you can based on these criteria, unambiguously distinguish between something that's an organism and something that isn't. So, with that, we're going to go to when does human life begin? And this will raise the continuum problem. I'm going to start with just a few slides of human development, since I realize that not everybody here lives, dies, and breathes, embryology the way I do, just to get you oriented. So this is a picture of a human embryo taken from a fertility clinic. You see a bunch of circles here, if my pointer will work, and it doesn't want to. There we go. This bright circle is the culture well that's maintaining the embryo. This sort of grainy looking circle in the middle is the one-celled human embryo, or zygote, and it's surrounded by sort of a clearer layer. This is sort of a jelly coat. It's called the zona pellucida, or the clear zone, and it's designed to protect the embryo during the first 5 days of life as it travels from the ovary down into the uterus and undergoes implantation. So this is going to be a time lapse showing you pretty much those first 5 days. So what you can see in this movie is that the embryo very rapidly will enter into a period of cell division, making smaller cells without growing in size. So just dividing up the mass of the embryo. These smaller cells are known as blastomeres. And after about 3 days, we reach the morula stage, because it looks kind of like a raspberry or a blackberry. And at this stage, the cells undergo a change in their properties so that they can adhere to each other more tightly in a process known as compaction. You can see the embryo sort of smoothing out a little bit. But the major thing that it does for the embryo is it allows the embryo to transport fluid to the inside, and it blows the whole thing up like a balloon, because the fluid can come in, but it can't come out because the cells are very tightly attached to each other. So after 5 days, we reach what's called the blastocyst stage, and you can think of this stage as being kind of like a fluid-filled soccer ball. On the outside, the black and white hexagons of the soccer ball are a cell layer known as trophectoderm that will largely go on to produce tissues within the placenta. And on the inside are a small group of cells known as the inner cell mass, or ICM, that is going to produce all of, or the great majority of the cells and tissues that you're carrying around with you today. Okay. So blastocyst is an important stage because it's the first point at which we have cells in the embryo that have distinct properties, and they're going to do different things. Everything goes pretty rapidly after that. These are pictures of real human embryos from the Condic collection. And what this diagram shows you is here's that one-celled embryo, which in reality is the size of the little dot over the number 1. And this is the first 8 weeks of human life. So this is the period known as the embryonic period. And it's the period over which you go from a single cell all the way up to a fully formed, albeit pretty small, this is only 5 millimeters, human body. So everything you have in you now, all of your organs, tissues, systems, and relations are produced in that short 8 weeks of time. The rest of human life in utero is the fetal period, where we're going to have a lot of growth and maturation of those structures, but we're not going to make anything new. So with that as a very, very quick overview, what is the continuum problem? And it's precisely this, that because development proceeds continuously from the one-cell stage up to birth and beyond, many people will set arbitrary points for when life begins and for when humans have rights. So a lot of people like this blastocyst stage, where you have the first 2 cell types arising. But you can go later than that. Many people like implantation, or the point at which the embryo first establishes a relationship with the mother. Many people point to a later event that occurs at about 2 weeks, where the earliest rudimentary structures of the nervous system arise in a structure known as the primitive streak. But you can go even later than that. A lot of people like this very ill-defined period called brain life, sort of analogous to brain death. So if brain death is when you die, then brain life would be when you develop mature patterns of brain function, somewhere around the 5th week. But again, you can even go later than that. Peter Singer at Princeton is famous for having posed self-awareness as the meaningful beginning of human life, an event that he asserts happens about 28 days after birth. But personally, as a developmental neuroscientist, given the very long period of time over which the human nervous system develops, this one's my favorite one, brain maturation, something that doesn't happen until 25 years after birth. So several people people in this room are probably not quite human beings yet, all right? Send you back to the human parts factory, okay? But I hope what these definitions show you is that choosing among them is merely a matter of utility and taste. I would also add power. Not a matter of science and reason. So how is it then that science addresses this question? Well, unfortunately, to answer this question, we have to ask two closely related questions. First, when does a new cell type get produced in the interaction of sperm and egg? And second, what's the difference between a cell and a human being, a person, an organism? So, thankfully, the first question is very easy to answer. Biologists use two very simple, straightforward criteria to decide when a new cell type is formed. Changes in cell type are detected by changes in material composition, what a cell's made of, so typically this is changes in gene utilization, or by changes in cell behavior, and very often those two things happen together. If you change what it is that the cell has inside of it as a toolbox, you can change what the cell can do. These two criteria are used throughout the scientific enterprise to determine when a new cell type has been produced, either in the laboratory or in an embryo. Of course, logic also requires that if a new cell type arises from the interaction of one or more cells, those cells have to disappear. So long as the human gametes that give rise to an organism are still existing, then we can't have something new. So based on these criteria, is the product of sperm-egg fusion or fertilization a new cell, or is it just a modified gamete? Many people call the product of sperm-egg fusion a fertilized egg. So is that a reasonable scientific term? Well, I hope to convince you it's a completely inaccurate and not at all reasonable scientific term, and that a new cell, in fact, forms at the instant of sperm-egg fusion. So this is a scanning electron micrograph of a much larger egg cell in pink and a little blue sperm cell, and in the process of fusing. So when these two cells interact, the outer surface of the two cells will adhere to each other. That's the outer plasma membrane of the cells. And it will fuse in a very rapid process that is well studied and takes about 250 milliseconds to complete. So about a quarter of a second. There is an instant where you go from two cells to one cell. The sperm in this process ruptures and it ejects all of its cellular contents in its nucleus. So clearly the sperm as a separate cell ceases to exist in this moment of sperm-egg fusion. Because the plasma membranes have fused, all of the lipid and protein that used to be uniquely present on the surface of the sperm is now free to incorporate into the surface of this greater combined cell, changing its molecular composition. So what about the egg cell? So following the fusion and destruction of the sperm, the resulting cell has a composition that's distinct from that of an egg. We've already talked about the incorporation of sperm-associated lipids and proteins into the surface of this cell, but the changes are much more extensive than that. The cell also has a new genetic composition, with half of its DNA being derived from mom in pink and half from dad in blue. But I think most importantly, or most tellingly, this newly formed cell also radically changes its behavior and acts in a very un-egg-like manner. So the entire structure and function of an egg is to find a sperm and fuse to it. And yet within 2 to 5 seconds of the fusion of the membranes of these two cells, this new cell will undertake a series of molecular events that over the next 30 minutes will result in the deposition of enzymes into that space surrounding the surface of the cell and that zona pellucida, that protein coat, protective coat. And the function of those yellow enzymes is to destroy all remaining sperm binding sites. So the entire raison d'etre of being an egg is to fuse to a sperm, but the very, very first act of this new cell is to antagonize the function of the gametes from which it's derived. So I would say the egg as a separate cell, as a haploid gamete, also clearly ceases to exist, and a new cell with unique behavior, unique material composition, and unique genetics is formed at this instant of sperm-egg fusion. So what is this new cell? This is our second question. So its scientific name is the zygote or one-celled embryo. But, you know, looking at this picture, even for me as an embryologist, doesn't really tell me much. Now, I don't look at that and go, oh, it's a baby. And it doesn't look like a baby. And that is precisely the problem. At early stages, we can look at a zygote or a morula or a blastocyst, and recognizable human form and function are not evident. So how do we know that this is a new human being and not just a human cell or a ball of human cells? Actually, this is a surprisingly difficult question to answer. And it's difficult to answer for at least 3 reasons, and all of these reasons will be very relevant to the question of death as well. The first reason is that cells themselves are living organisms. So cells exhibit autonomously coordinated activity of parts. They're made up of parts, and those parts work together for the good of the thing as a whole. So I'm going to show you this little movie. This was made in 1958 at Vanderbilt University. Investigator took some of his own blood and put it into a culture dish, and what you're seeing here are three players. So the star of the show is this cell in the middle. This is a neutrophil, and its job in the body is to seek out bacterial invaders and eat them. Then you're going to see these little round cells that are blood cells, and they have lots of jobs in the body, but they don't do much in a culture dish. And then you'll see these little dots, which are bacteria. So here you can see the cell has activity. It coordinates parts. It can sense its environment. It can avoid obstacles. It's clearly chasing those bacteria, trying to get them. So it's acting like it would in the body. The suspense is killing me. Is it going to get it? Yes. Okay, so this is a little creature. It's a little one-celled organism, but it's also a cell. It's not a person. So cell type specific properties and functions will persist in vivo. So this cell is doing exactly what it was built to do when it was part of the body, but it's no longer part of the body. Okay? Second reason it's hard to distinguish between cells and organisms is that cell-cell interactions can mimic many naturally occurring developmental processes. So this, again, is a scanning electron micrograph. It's of a mouse embryo at about, let's say, E10 maybe. What do I say there? I don't remember how old it is. Probably around 10 days of development. And it has these little structures here that are known as limb buds. So they're the beginning formation of the legs and the legs of the mouse. In us, they would be either legs or arms. So this group of investigators was interested in how much normal development could we see just in a culture dish. So what they did was they chopped off limb buds from a mouse embryo at about day 10.5 and placed them in organ culture. And that's what you're seeing in this picture. So at this stage, limb buds are very, very simple structures. They consist of 2 cell types, an epithelial cell layer on the outside and a loosely organized, undifferentiated group of cells called a mesenchyme on the inside. So they maintained these limb buds for 7 days, and this is what happened. This middle picture shows the overall structure of what formed, and these are the bones that formed within that limb bud. And this is just a cartoon depiction of what a natural skeletal structure of a limb would look like at a comparable age. So clearly, this limb bud all by itself can undergo many of the natural processes of development in a tissue culture dish. The last is that even more complex systemic functions can arise and persist in organ culture. So here was a study of wound healing. In this, they took some fetal skin from an animal, wounded it in culture, so they made a gap in between 2 sections of the skin, here and here, and waited 72 hours in culture, and it healed itself through a process that was almost identical to what would happen if you scratched yourself in your body. Even more complex than that, here's a little bit of a gruesome experiment. These researchers took a bovine liver and a kidney, and they put them together in a normal systemic relationship to each other. They plumbed them together with plastic tubing and ran pumps to circulate the blood. And many characteristic functions of these 2 organs were maintained, including homeostasis, fluid balance, urine production, ion balance, and blood pH, detoxification of the blood. So all the things that those organs would do in your body, they could do in this completely artificial circumstance. So complex systemic functions can persist in culture with sufficient technical support. And this raises the central problem, thinking about the beginning and end of life. Both human cells and human beings are alive, and they're both human, but human beings are not just a collection of living human cells. So how do we tell the difference? And if you take home nothing else from this lecture, remembering that this is the central question, and, challenging your own understandings to address this question is probably the best thing you can do for your development of thinking, responsible citizenship, taking care of an elderly person who's dying, or, God forbid, a child who's in the hospital. So to answer this, I think we have to digress a little bit into theology and philosophy. What causes a human being to be different from a cell? So the church teaches that the rational soul is the unique form of the body. This was articulated in the Council of Vienne in 1311, the Fifth Lateran Council in 1512, and a brief by Pius IX in 1857. The Catechism of the Catholic Church states, the unity of soul and body is so profound that one has to consider the soul to be the form of the body. That is, it is because of the spiritual soul that the body made of matter becomes living human body. Spirit and matter in man are not two natures united, but rather their union forms a single nature. What does this mean for our understanding of the difference between cells and human beings? The substantial form or the soul causes all acts of a living being. That's what it means to be the form of the body. So the soul is the organizing principle of the body. It causes all bodily activities, including the very life of that entity. And all living beings, if we follow upon Aristotle and Aquinas, all living beings have souls. Bacteria have souls. Plants have souls. Anything that's alive has to have an organizing principle that causes it to be alive and to function as a unified whole. The human soul is intrinsically united to the body, and all acts of the soul manifest as bodily acts. So even when you're thinking, there's going to be activity in your brain that reflects the fact that you're thinking. Cells removed from the body, or after death of the person, undergo a substantial change, and they obtain a new kind of soul, a cellular soul. So that little neutrophil was not being controlled by the soul of the investigator in the culture dish, just because it had originated with him. It goes from being a part of him to being a thing in itself. And the nature of that thing is determined by the developmental processes that went on when that investigator was an embryo, that built into that cell its properties. But the cell becomes an autonomous actor once it's outside of your body. So, levels of organization. Going from non-living organic matter, moving up to the next level of cellular organization, like that little neutrophil, requires living cells. And cells are able, like organisms, to integrate all of the functions required for life. Moving up to, say, tissue, for example, that little limb bud that we discussed, can be explained by short-range interactions generating complex structures. So the intrinsic properties of those two cell types within that limb bud were able to build, through their interaction, bones and joints that were natural to a limb. Moving up to the next level requires a living being, or an organism, a multicellular organism, where all of the cells function together as an integrated whole for the good of the thing as a whole. Now, the human soul orders all the properties of the organism and all of its cellular parts. But the molecular composition of individual cells that have been removed from the body determines their behavior. This would be the argument I would make. And it's important to reflect on two very profound intellectual errors when you were thinking about these topics, that of reductionism, both of which are rejected by the church, and dualism. So a reductionist, this is what most people think of the scientific perspective as advocating. A reductionist would say that a human is just a collection of cellular parts that all add up to a living being. And development is a process that involves gradual acquisition of complex cellular function, and therefore there's no single moment at which life begins. That graded series of you pick wherever feels good to you. And when a human can be considered alive enough to be the subject of human rights is an arbitrary decision. In contrast, the dualist on the opposite end of the spectrum would say that the soul is not united to the physical body, it's somehow a ghost in the machine. You have this spiritual essence, but it doesn't have to be reflected in actual acts of the body. So therefore, the soul can have immaterial acts that are not associated with any physical acts. And so long as there's a potential for development, the soul might be present. And consequently, gametes or stem cells or other entities that show some similarity to embryos could be humans, and we just can't know. So both reductionism and dualism support the view that the precise moment when human life begins cannot be determined by science. And I therefore reject both of these views, because we can determine this. And in order to determine it, we have to focus a little bit on the difference between a multicellular organism like us and that little neutrophil in the culture dish. So multicellular organisms such as human beings are uniquely capable of development. The self-directed maturation to a characteristic adult form. Interactions between subcellular and cellular parts are ordered to the good of a multicellular whole. They don't function just to keep those cells alive, they function to build the body you're ultimately going to need as an adult. Adaptation and wound healing exist to preserve the healthy function of a multicellular whole rather than the healthy function of the individual cellular parts. So, based on that, I think we can ask, when does organismal function begin in development? And I think most everybody would agree this is an organism. It has parts. Parts work together to support the thing as a whole. I think most people, even some of our opponents in the pro-life movement, would say this is probably an organism. It has parts, and those parts work together to support the life of that thing as a whole. But is this an organism? So I have a friend who's an attorney, and every time he sees his picture, he always insists, nah, that's not an organism. It's a bare aspirin. And I have to admit, it kind of looks a little bit like a bare aspirin. But I hope to convince you that from the one-cell stage onward, human embryos function as human organisms that are autonomously producing more mature multicellular human form. Okay, so why do I say that? We're going to go back to the features of organisms, starting with development. And development is the entire business of an embryo. This embryo enters immediately, within seconds, into a pattern of molecular interactions that can only make sense. Not to keep the cell alive or to keep it happy as a single cell or as a group of cells, but only make sense relative to the nature of development. We block sperm entry, additional sperm entry, because at this early stage of life, if you have more than the normal 2 copies of DNA, that is incompatible with development. And that's not incompatible with life. Many cells in your body, for example, all of your muscles, have multiple nuclei and, dozens of copies of your genome, and they don't care at all. So the embryo is not doing it because that's necessary to be healthy as a cell. It's doing it because it's necessary to develop normally. And that's the very first act that this cell takes. What about autonomy, or the repair of injury? Embryos are actually quite good at this. I'll give you just one example, that of pre-implantation genetic diagnosis. So what you're seeing here is an embryo that's been immobilized by suction against a smooth pipette, large pipette, and a smaller, sharper pipette is inserted to remove a single cell. So this is typically done in assisted fertility procedures to determine if that embryo is genetically normal, if either the parents perhaps are carriers of genetic disease or are older. Typically, they take a single cell from an 8-cell embryo, and the biopsied embryos are allowed to develop into the blastocyst stage, and if they pass the genetic test, then they are transferred to the uterus. Now, think about it. This can only be described as a catastrophic injury. This embryo at a tender age of 8 cells loses 1/8 of its body. Okay, that's a pretty serious, injury to recover from, and yet they do. Embryos will regenerate the missing tissue, and such that there have been many tens of thousands of live births from biopsied embryos worldwide. What about adaptation or adjustment to changing environmental circumstances? Well, again, embryos are pretty good at this. I'll give you just one example. Ectopic pregnancy. So in most cases of ectopic pregnancy, the embryo does not proceed to live birth because it implants in a location where it can't establish a placental circulation with the mom. And we are very dependent on that circulation. We don't have a lot of nutrients as early embryos. We're not like chicken eggs where there's a lot of food for the embryo to eat. So if we don't make that circulation connection with mom, not going to make it. But there are cases where embryos implant in completely abnormal environments, and they do just fine, like this case in 2003, where this little baby, Nahalanisi of Philippi, South Africa, developed 9 months entirely within her mother's liver. Whoa, how did that even happen? Okay, so she was ovulated from the ovaries that are up here, and she should have gone down here to the uterus, but she took a wrong turn and ended up in her mother's abdominal cavity. And she was fortunate enough to attach to the liver, which is a very soft organ and also highly vascularized. So she established a normal placental circulation through the hepatic veins of the liver. And at the end of 8 months, I can't even say they did a C-section. They just cut the mom's abdomen open, cut a slit into the liver, and popped the baby out, and both baby and mom are perfect. Now, if a baby can develop 9 months inside a completely abnormal place like a liver, I think that's good testimony to the adaptive capabilities of embryos. So what about the last one, integration? Again, embryos are very good at this. This is a little cartoon. I'm just going to give you one example. Summarizes an enormous amount of work that demonstrates that the pattern of cell division at the 2-cell stage will determine what structures are produced from each of the cells at the 4-cell stage. So in a pattern like this one, the pink cells and the red cells are going to preferentially contribute to, though not exclusively, to the postnatal body by finding themselves within the inner cell mass. And the green cells and the blue cells are going to preferentially contribute to the placenta. So this could happen a lot of ways, but we know how it happens from an experiment like this. If you take 3 embryos that are all showing the same pattern of cell division, and from each embryo you take 1 red cell, 1 blue cell, and 1 green cell, and you take those cells of the same type and put them together and transfer them to a surrogate mom, what you find is that in 85% of the cases, the red cells can make a healthy mouse out of themselves, but in none of the cases can the green cells do it all by themselves. And what that tells us is that even at the 4-cell stage, cells have different developmental abilities, and that they normally have to work together to produce a healthy animal. So I wrote a review of this in a Journal of Law and Public Policy in 2014, and if you just search my name and When Does Life Begin, it's a free paper. So again, if you want to do a deep dive, this paper summarizes hundreds of citations from the primary scientific literature and many, many events occurring early on that can only make sense as the integrated activity of something that's working for itself. So it's pretty clear from this review that sperm egg fusion sets off a molecular cascade that controls events that will take place hours or even weeks later in development, and that these events are not required for healthy life. They're only making sense as part of a developmental sequence produced by an organism. And while some scientists may dislike or deny this conclusion, it's supported by hundreds of peer-reviewed scientific papers and is entirely uncontested in the literature. Embryos are developing organisms. I mean, I've given this talk or a version of this talk many dozens of times, and I've always said to any scientist in the audience, hit me with your best shot. You know, if there's a paper out there I'm not aware of, bring it. And you can find lots of papers where people will assert, oh, no, nobody knows when life begins, but not a single one that presents even a shred of data that contradicts this conclusion. So what can we conclude about the beginning of human life? A new cell, the zygote, with unique material composition, genes and behavior comes into existence in this instant of sperm egg fusion, and it exhibits all the characteristics of a full, complete, and albeit immature human being. And therefore, human life commences with the onset of organismal function at sperm egg fusion, or the one-celled little bear aspirin is in fact a human being. And it's an evidence-based definition of when human life begins. So don't let anybody tell you, I didn't mention Revelation or Scripture or my own personal opinion once. This is what science tells us. Okay, so now we're going to take the opposite end. of this problem. When does the human life end? So thinking about death is very challenging. I used to, for years, do the talk I've just given with maybe one slide on death just to kind of make the point of it's symmetrical. You apply the same criteria at both ends. And at the end of the talk, 50 hands would go up, and 49 of them would be questions about that one slide. Okay, so I think we all passed the embryo stage. And we can say, however that happened, it happened, okay? But we're all facing death. It's a lot more compelling to people. So it's difficult. There is not a universal consensus among Catholic thinkers on the criteria for death, and there is no official church doctrine. So therefore, you're free to think whatever you want and hold your own opinions on this matter. However, appreciating the differences between human cells and human organisms, I think, is critical to making a sound judgment. And I will admit that this is a work in process. This has been one of the tougher things I've had to think about professionally. So what is the current definition of death? Now, I will note, as I did at the beginning, there are efforts underfoot to change this definition to include a much looser set of criteria. But the current Uniform Determination of Death Act, which is approved by the American Bar Association in 1981 and is supported by legislation in all states, states an individual who has sustained either 1, irreversible cessation of circulatory and respiratory functions, or 2, irreversible cessation of all functions of the entire brain, including the brainstem, is dead. And the preamble clarifies that under Part 2, the entire brain must cease to function irreversibly. The entire brain includes the brainstem as well as the neocortex, or the thinking part of the brain. The concept of entire brain distinguishes determination of death under this act from neocortical death or persistent vegetative state, and these are not deemed valid medical or legal bases for determination of death. So I think right now we have a pretty good definition of brain death. That's not to say it's not without problems. So I'm going to go back to this levels of organization and what's required. So what does death require? I think the most intuitive and simplest view is that death requires a loss of all organization or total disintegration of the living being. And this idea of total disintegration actually has pretty strong precedent, that of Saint Pope John Paul II, who stated, the death of a person is a single event consisting in the total disintegration of that unitary and integrated whole that is the personal self. It results from the separation of the life principle or soul from the corporal reality of the person. But what exactly is total disintegration? And what does it look like? And how do we determine when it's happened? Okay, so we've really got to bring it down a little bit more to brass tacks here, because that could mean a lot of things. Okay, what total disintegration doesn't mean? Death is not the complete loss of bodily unity or total disintegration in the simplest sense. So we don't immediately turn into dust, when we die. Wouldn't that be great? We would have no ambiguity whatsoever. A living person, bam, pile of dust. Okay, but that just doesn't happen, okay? God, if you had just consulted me, before making the universe. Okay, death is also not the complete loss of cellular life. We don't instantaneously turn it into a decomposing corpse. It takes a long time, actually, for the body to decompose. Thirdly, death is not a complete loss of cell communication and bodily structure. We don't suddenly disintegrate into a pile of dissociated cells that we, like sheep, are all going to go their way, right? That doesn't happen either. Okay, so we don't turn into dust, we don't turn into a rotting pile of organic molecules, and we don't turn into a pile of disaggregated cells that all goes its willy-nilly way. So what is this total disintegration? What survives after a declaration of death? Okay. Well, one of the most disturbing things that survives is living cells can survive in a corpse for a very long time. So I didn't even know this until I started looking into this, but there are journals called things like Journal of Forensic Science, where they literally go and find dead animals that are usually animals that have some valuable genetics to them. They're breeding animals, and they want to preserve cells so they can clone those animals. And then they determine how long can they get living cells out of this dead animal. And it depends on temperature, not surprisingly, but this is days on the bottom here. Up to 60 days at 23 degrees. And this is the percent of viable cells that they obtained from the samples that they took out of the corpse. 60 days is a long time. A little bit shorter if you warm the corpse up a bit. But still, 30 days. A month. So cells can survive and continue to function for weeks after declaration of death. There is a period of rapid cell death, particularly for cells that are very oxygen dependent, like cells of the brain and the heart, where they're going to die off very quickly. And eventually there'll be loss of all metabolic function, and we will eventually get to a state of decomposition if you just let the corpse sit around long enough. Hours, days, or perhaps even months. If you supply external life support, you can extend this. Just like that neutrophil in culture, you can keep cells alive. But eventually you get to decomposition anyway, in most cases. Though there are rare and unusual exceptions, which we will turn to next. So a very good man, Alan Schumann, who's a neurologist, wrote a number of very, very important papers in the late 1990s about brain death. And he identified a number of functions that persist in the body after death of the brain. Now, Schumann is of the belief that brain death is not real death, that people are still alive after their brains irreversibly, their whole brain ceases to function. And he bases that conclusion on things like this, that you can see these functions in a person or in the body of an individual who has sustained total death of the brain. He acknowledges that all of these functions are significantly impaired after death of the brain. But how are we to interpret this persistence of function? Is the person really not dead? And the interpretation is complicated by the fact that most of these functions are preserved at least to some extent in tissue culture. So, immune defense. Well, isn't that neutrophil part of the immune system? And does its function persist in a laboratory dish exactly like it would in the body? Absolutely. How about proportionate growth? Well, didn't that limb bud show proportionate growth to make something that looked an awful lot like a regular limb? What about wound healing? Didn't that skin heal itself in culture? What about homeostasis, elimination, detoxification, energy balance? Well, all of those can happen with two organs plumbed together with plastic. And on a more detailed level, if you look at, for example, immune regulation, this is just this is not even the complete way the immune system is regulated, but it shows the major players in the CNS in yellow over here, and in the periphery or the rest of the body in blue, and all the ways that they communicate with each other. So clearly in life, the brain plays an important role in regulating or integrating immune function, both receiving and sending signals from the body to balance the immune response. After brain death, immune cells will continue to function, just like that neutrophil did, as cellular organisms with the properties that were built into them in life, to fight infection in response to local signals. But there's no body-wide communication, and therefore there's no adaptation to changing external circumstances. So functions persisting after death of the brain are all significantly impaired. I think the easiest way of understanding this is there have been a number of studies looking at women who were pregnant close to the time of viability of the fetus, who suffered brain death, and their families made a decision to maintain that person on life support long enough for the fetus to mature so it could be delivered and survive. So this is a review of 30 cases of fetal gestation following death of the brain in the mom. And this is what the authors say. During extended life support, patients developed several complications, including infection, hemodynamic instability, diabetes insipidus, Penn hypopituitarianism, hypochondriothermia, metabolic instability, acute respiratory distress syndrome, and disseminated intravascular coagulation. The authors characterize the, quote, enormous challenges of management and offer guidelines for continuous active adjustment of cardiac output, respiration, endocrine balance, body temperature, thrombosis, nutrition, as well as standard fetal monitoring. So these women were kept alive by extensive minute-to-minute regulation by a medical team. They were not keeping themselves alive. So after death of the brain, most body systems function only in very limited range and do not autonomously adapt to changing circumstances for the reasons that I showed you on that immune slide. Once the brain is out of the picture, information that's extended to those systems is very, very limited. So the doctors have to do it. There was even a more recent paper a few years later, in 2021, that looked at 5 additional cases and drew the same conclusions. So there are 2 views of death that are opposed to Catholic teaching, not surprisingly, reductionism and dualism. And the arguments are very, very analogous to those about the beginning of life. So the reductionist would say a living human being is a collection of cellular parts that add up to a living person. And death is a process that involves the gradual loss of cellular function. Therefore, there's no single moment of death. And when a human being can be considered dead enough, like we saw in that very first slide, we can debate whether they're dead enough to be dead for organ donation, and it's an arbitrary decision. The dualist, on the other side, would say the soul is not united to the physical body, it's a ghost in the machine, and therefore the soul can have immaterial acts that are not associated with any physical acts. So long as the body persists, the soul might still be in there. And consequently, you can't be certain the soul has left until the body, in the entire bodily system, breaks down. And both reductionism and dualism support the view that a person might still be in there after declaration of death. And we just can't know. So that brings me back to science again. Scientific parsimony is a very entrenched concept in the profession, and it's a way of framing this discussion. So if a biologic function can be explained solely by the properties of cells, then a higher level of organization should not be invoked. I don't need to imagine that the soul of that researcher with the neutrophil is reaching out into the culture dish to control the behavior of that cell, because I can explain the behavior of that cell based on the properties of the cell itself. If a biologic function cannot be explained solely by the properties of cells, then a higher level of organization must be invoked. So the reductionist would assert that number 1 is always the case, and the dualist would assert that number 2 can never be ruled out. So can we find, again, that golden mean in between these 2 extreme positions? Bringing me back to the one thing I want you to remember from this talk: both human cells and human beings are alive and human, but human beings are not just a collection of cells. So how do we tell the difference? And I think the answer to this with respect to death is, are there any biologic activities that cannot be explained simply by the properties of cells? And I would argue that persistence of a rational animal must be due to a human soul. That we don't just add up enough human parts and get a rational animal out of it. So humans are rational animals, therefore clear evidence for the persistence of a human soul can be provided either by rationality, or what I will call animality, or organismal function, functioning like an animal, being able to pull all your own activities together and make it work. And this gives us 2 clear criteria for a living person: persistence of the capacity for mental function, no matter how impaired. Mental activity requires a functioning brain. Or persistence of intrinsic autonomous integration of bodily function, being an animal, even in the absence of evident mental function. And both of these functions are due to the same organizing principle, and therefore the persistence of either one of them is sufficient to conclude to the persistence of a human soul. And I'll give you just 2 examples. There are really hard cases, and if I had an hour and a half, I would go into some of the hard cases, but I'm not going to do that to you. Everyone remembers Christopher Reeve, famous actor, had a horse accident, was a high cervical ventilator dependent quadriplegic. He was conscious. and remained conscious for a long time. He had limited autonomous integration, so similar to those brain-dead women who were gestating a fetus, he had to have daily medical intervention just to keep everything going. He was dependent on life support. He couldn't breathe on his own. I would argue he no longer functioned as an organism. So that raises a lot of really interesting philosophical questions, and anybody out there with philosophy background who wants to debate me, I'd be happy to hear your opinion. But he continues to have mental function. And based on that, I would say he's alive. How about Terri Schiavo? So she had limited or absent consciousness. She sustained autonomous integration beyond food and hydration. She balanced her own life, was able to maintain herself with minimal intervention. Normal care, food and water, not life support. She no longer exhibited any kind of mental function, and the autopsy after her ultimate death of her brain supported that organ was probably not very functional, if functional at all. But she continued to function as an organism. She continued to function as somebody who was comprised of parts and who maintained those parts on her own for the good of the entity as a whole. And therefore she was still alive. She was in a very bad state, as was Christopher. So how are these situations different from brain death? So after death of brain or cardiac death, all 4 marks of a living being are absent. So vital functions are no longer autonomously maintained. No internal system exists that can respond to the needs of the body as a whole, so integration is lost. There's no development. There's never been a case of an individual undergoing normal development after death. And there's only limited detection of both internal and external conditions, and therefore no systemic adaptation. So going back to our levels of organization and the idea of total disintegration, living cells and tissues are not the same as a living person. And therefore death does not require moving all the way down to organic matter. Moving down one step is death. And I'm perfectly comfortable with a dead body showing certain kinds of biologic activities that are dependent upon the function of the cells within that body. Now there is one caveat, and I'll end with this, that diagnosis can be a serious problem. So we've all seen headlines like this: brain dead student wakes up hours after doctors suggest pulling the plug. He certainly doesn't look dead in this picture. Some individuals are referred to as brain death by those who advocate letting the patient die. So doctors can be quite loose with their terminology. And this individual might have sustained a serious brain injury, and maybe the doctors didn't feel it was worth investing the money and keeping him alive, so they called him brain dead. And then there are cases of pediatric patients. This is Jahi McMath, who were declared brain dead by standard medical criteria, and yet they continued to exhibit behavior and functions that contradicted that diagnosis. And ultimately, I think the consensus of the medical neurological profession was that she was misdiagnosed. And it's a challenge with pediatric patients in particular. Kids can sustain a lot more injury than we can and not be dead. So how do you know with certainty that there has been irreversible cessation of all the functions of the brain and the entire brain? So I'd say the standard way of assessing death is by clinical criteria, but there are also functional criteria. So this is a MRI of a brain showing healthy brain structures, and this is a kind of scan that shows metabolic activity within that brain. So warmer colors are more activity, cooler colors less activity. Here's a scan of an individual who came in after a stroke. This was the first scan that was taken for this patient, and you can see an area of her brain has no activity at all. She was maintained for a couple of days and then rescanned, and this is the second scan. So this is her skin and hair, and this is her brain. So that's no activity. That is a dead brain. You can also look at circulatory flow to the brain by a less invasive and or less expensive test of looking at the supply of blood and oxygen to the brain. And in cases of brain death that's due to occlusion of the vasculature, it's a pretty unambiguous answer. This brain is not getting any oxygen. Brain cells are very dependent on oxygen, and within a few minutes they'll be dead. Okay, so concerns about misdiagnosis, and I'm nearly done. Advocate for revision of pediatric criteria. I think this is one of the most serious neurological medical problems we have today with respect to death, is that we don't have good criteria for children. Be educated and ask questions. Do what you're doing today. Invite people like me to come talk to you. Get a second opinion. You don't have to take the first doc who comes along and says, oh, your child has brain death. Rely on a functional diagnosis if you have that as an option, and if they aren't offering it to you, insist on it. Wait. That lady who had a stroke, within 48 hours, it was very unambiguous that she was dead. And consider the tough questions in advance so that you're not going to feel pressured, either by a doctor who's wanting you to make a decision that's in the financial interest of the hospital, by relatives who have your welfare at heart and don't want to see you suffer. Think about it. Ask, is providing food and water life support? What if we do nothing to prevent death? What are extraordinary needs? So conclusions regarding death. Death never involves instantaneous total disintegration of all biologic activity. Many biologic functions can persist after declaration of death by any means, both with and without life support. Yet all of these functions are significantly impaired after brain death or cardiopulmonary arrest, and most are also observed in cell and organ culture. Two reliable signs that a human organism persists are the persistence of self-directed integration and the persistence of any form of brain function or the possibility of brain function, even if we don't have good evidence for it. So the human soul controls all the activities of life. Autonomy, integration, development, and adaptation are the unique hallmarks of a living multicellular organism. Organismal function begins in that little bare aspirin at sperm egg fusion. Cells removed from the body or persisting in the body after death retain characteristic properties with interactions among different cell types producing interactive biologic structures and supporting complex functions. But that does not mean the person's alive. Persistence of either organismal autonomy or brain function provides good evidence that a human being is present, and a sound view of both the beginning and end of life must explain the difference between a living being and living cells while avoiding the errors of dualism and reductionism. So I hope I've made this incredibly clear to everyone, but chances are, it's a complicated question. If you have questions, I'm always very, very happy to entertain email questions, days, weeks later. So please write down my email and feel free to reach out to me if I can help in any way, but I will take questions now.